Her Majesty, welcome everybody. We are having our next 3 talks from researchers from our network, CIBERNED. So the first one will be by Jesús Ávila, and the title is The Effect of GSK3 Beta Overexpression in Adult Neurogenesis. Please, Jesús. Thank you.
Okay, thank you. It is well known that one of the main features for Alzheimer's disease is the loss of episodic memory. Everybody talks that it's a loss of memory related to Alzheimer's disease. This loss of memory could be related to a deficient adult neurogenesis that takes place at the dental gyrus in the adult. This adult neurogenesis takes place from the first proliferation of some stem cells that start to proliferate and to differentiate into new neurons, and these new neurons could connect with the neural network, and the function of this new neural network is related with memory and learning. Also, you know that in Alzheimer's disease, there are 2 histopathological features: the appearance of the beta amyloid that could aggregate into senile plaques, and the presence of tangles that are composed of tau protein. Then, it has been from the beginning of the studies of Alzheimer's disease to try to look for possible connections between these histopathology features and the loss of memory. I mean, if amyloid or tau could be related to the loss of memory. It's well known that there is several connections between amyloid and tau, but the connections between tau amyloid and memory is less known. One connection between amyloid and tau is through one enzyme that is known like JSK3 beta, mainly because it's the form that is mainly in the brain. We know that amyloid activates this enzyme, and once this enzyme is activated, could phosphorylate tau. In fact, this enzyme is also known like tau kinase 1, and this tau that is phosphorylated is toxic. Then the thing is, this toxicity could be related to some changes in neurogenesis and to changes in memory. Then the objective of our work is to know the consequence of overexpressing JSK3 before and after neuron differentiation during adult neurogenesis. And to do that, we use several models, mice models, conditional models, that we can overexpress the enzyme or downexpress the enzyme using 2 promoters that express the enzyme. One promoter is expressed before neural differentiation, the other one is expressed after neural differentiation. One promoter is expressed in this type of cells, stem cells, and the other one is expressed probably from here. To test if it really is expressed from here to the differentiation, we tested by immunofluorescence studies the presence of a specific marker for the different types of cells and to see when the reaction against the overexpressed JSK3 appears. And we found that the overexpression from this point and so on continues in the adult neuron. we try to connect this with the loss of memory, as I told you before. And then the first type of experiments that we did is to test if it's a memory impairment due to the overexpression of JSK3 in these conditions. To do that, we use two behavioral methods for the mice that was theoretically with memory impairment. One is the Morris-Water maze that, as all you know, deals with the mouse should remember where is a platform when it's through to this swimming pool, and after several days, the mouse knows perfectly where is the presence of the platform. But in our case, the mouse that is normal, wild type, is doing better than the mouse that is overexpressing JSK3. Then, in this case, it's some kind of memory impairment. Also, but using other tests for memory, the object recognition test, we found also that there is a memory impairment in this type of mouse. Then we have a mouse without memory. And when you lose a function, usually you look if you also lost a part of the structure in the brain. It's something damaged in the brain of that mouse. And in our case, we were looking to the brain of the transgenic mouse, the one is overexpressed in JSK3, and we found that this part of the brain, the dental gyrus, is degenerated a lot compared with the thing that happens in the control wild-type mouse. Then the damage is here. There is less dental gyrus. The decrease in dental gyrus could be due to two things. One thing could be to the death of neurons, and the other thing could be because in this place there is adult neurogenesis, or the formation of new neurons, could be due to the absence of those new neurons. To see if it's a problem of dead neurons, the death, we found that JSK3 could modify proteins like beta-catenin that is a neuroprotector. If it's phosphorylated, it's degraded, and it's no longer neuroprotection. If the other one is that could be phosphorylated is tau protein. And tau protein, when it's phosphorylated, is toxic. The consequence in both cases could be death of neurons. But we were more interested in to know what happens about the adult neurogenesis. To do that, we try to look for new form neurons. And in this case, we use a virus that could express a green protein, and that is only infecting proliferating cells. When we did that, we can infect the proliferating cells and wait several times to wait until these proliferating cells became new neurons and adult neurons. Then, when we infect the dental gyrus, we can find the appearance of new neurons that are colored, green neurons, and then we can compare the appearance of these neurons from the wild type that has this form, Y shape, with the shape of the conditional transgenic mouse that has V shape. Then, to do like this, the Anglo-Saxons, to do a long story short, I will tell you the consequence of this. The V cells in the transgenic mouse are not functional neurons. These functional neurons are dead in a short time, and the consequence after the death is microglia is activated, and after activation of microglia, there is secretion by these cells, microglia, of new compounds that could be toxic for the precursor of neuronal precursors. This is indicated here. That is the activation of microglia, and microglia could secrete some new compounds that could kill the neural precursors. If we have less neural precursors in the niche, we will lose the possibility of new neurogenesis. From the molecular point of view, we are now in the middle of the experiment. We know that JSK3 could phosphorylate another protein, NF-kappa B, that could act in phosphorylating form, like a transcription or co-transcription factor that results in a decrease in the amount of a protein called fractalkine that is involved in this change in microglia. And the result in this change is that there is a protein, eotaxin, that will appear afterwards that is increased as a consequence of that. Then, this is a model in theory for Alzheimer's disease. If there is an increase that it could take place, and apparently take place, in the patients of Alzheimer's disease due to an increase in JSK3, we can start to protect, if it's the cause of the disease, using an inhibitor of JSK3. Like we have a conditional mouse and we can express up or down when we like, we can try to know what happens when we decrease, like we use an inhibitor, this overexpression of JSK3. We have reversion, we have the recovery of the memory of the mouse when the mouse is down, but we were unable to recover the memory when it's very old. And the explanation for that is that if we have a depletion of new neurons, because we have a depletion of neural precursors, as I told you before, then we were enabled to do a recovery of the memory. Then it's the thing that happens when we use a promoter, the one that was expressed late during the neurogenesis. What happens when we use the other promoter, the one that is expressed early in the stem cells? In this case, we found first that is expressed there in the stem cells, and the consequences are yet the opposite. In this case, we have a larger dental gyrus, and the mouse, or the mice, saw better memory using different paradigms. Then the conclusion I don't know, this is not going. Oh, okay. The conclusion is what happens in the human being, because we have the two differences. Depends when we overexpress JSK3 at the beginning or at the end of neurogenesis. To know what happens in the humans, we tested using the Golgi, Cajal staining, and we found that it seems similar to the first case, when the JSK3 is overexpressed but under calcium calmodulin kinase promoter, and we have in humans cells with a V form. Then we are discussing, and is the conclusion in this case, if we need to use inhibitors for JSK3 or not, to try to use like a target for the disease. For the moment, we think that probably it's not very good because it has another collateral problems. Usually there is no regeneration in peripheral neuron cells and another kind of things. But before end, if I can, and as suggested by Professor Kachaturian, that is looking for new features, for new ideas, or for new things, I will like to only to focus you on one thing. I told you about eotaxin, that eotaxin is something that was increased in all this story. And eotaxin is an aging factor. And you know, all of you know that aging is a main risk for Alzheimer's disease. And this protein is now very famous in some way, or is a fashion protein, because there are several relevant papers in the so-called the vanity journals, those with the very top impact index, indicating that when you I think that the battery is going down. Okay, it's very low. I think that the next speakers will have a new battery to avoid problems. There is something called parabiosis that is to change the blood from a young to an old mouse or vice versa. And the experiments indicated in these papers indicate that when you put blood from young mouse to an old mouse, there is a dramatic increase in neurogenesis in the adult old mouse. And when you look which component of blood is involved, you can see that there are several, like beta-2 microglobulin, but mainly is the one that we just told you is eotaxin. And yes, also to follow a little the talk of the Professor Rosor, he told us that to prevent the disease, there are a clinical trial in Colombia indicating that try to prevent the appearance of Alzheimer's disease in familiar cases. In familiar cases are those cases in which you have a mutation in a gene and you know that with that mutation, if you live enough, you will have the disease for sure. But not only that, they know that with this mutation in one gene, preselinin 1, and a specific that change a glutamic acid by an alanine, you know that the appearance of the disease is at 44 years plus minus 2. We know even that. Then when they were studying these cases in Colombia and Antioquia, is the thing that Professor Rosor said, they found another part of the family showing that was another population in which the appearance of the disease was 50 years old plus minus 2. It's 10 years later. Then it's the case that should be a protector factor. And when they did a genetic screening to see what happens with these people that develop the disease later, they found that the main change was in the gene of eotaxins that is going down the function of this gene. Then this could be some kind of example of looking new ways to try to at least to delay, I don't say prevent, but at least to delay it and to look for the appearance later of the disease, and that could be enough probably for the moment. And finally, I think that this is the people that did the work, all these people, Feliciana, Maria Llorens, mainly Maria Llorens did a lot of this, and Jerónimo Jurado in this case. And also part of this work and another work was done in collaboration with many groups of CIBERNED. I have some, probably I missed someone, but we have collaboration with more than 15 groups of CIBERNED until this moment. Thank you, I finished. Thank you.
Do you you hear me? Yes. Yes, please.
I found your data on the different effects of the GSK3 beta inhibition in young and older mice in terms of the neurogenesis very interesting. How much of those effects do you think can be attributed to phosphorylation of tau? Has anybody looked at those types of manipulation in tau null mice?
No, sorry. There is not only phosphorylation for tau. Yeah, there is also side effects, like these collateral effects. There are phosphorylation in different places of tau, but beta-catenin is also phosphorylated, as I told you. And probably we were looking for another microtubule proteins or related proteins. One of them was LC3 that you know very well. But that is something that we don't have until now a very good and real picture. Probably I will need to have another year to tell you the final picture of everything.
Any more questions?
Is GSK activated only in subgranular zone or also in subventricular zone? In other words, is activation of GSK related only with neurogenesis and memory, or in general with neurogenesis?
No, I think it's a good question. I think I only focus in dental gyrus, and this overexpression is only with an isoform of GSK3 that is GSK3 beta that is mainly expressed in brain. But we need also to study what happens in other places where this CAM kinase promoter is expressing. And that is expressing also in cortex. And we think that perhaps in the other neurogenesis place, that is the subventricular zone, could be also something that could take place there. But in this case, we mainly focus in the dental gyrus.
Any more questions? I have one myself, Jesus, just because you brought out the idea of the parabiosis. I think this has to do a bit with an ethical problem maybe, but do you think that parabiosis could be tested in humans?
No, I don't think so, and I think that it will be terrible. If anybody from here said that the blood of the young people could cure the old people, and in one case there is poor people, and the other case is rich people, could be a disaster. Then I don't think that this is the way. I think that it's better the other way. Once that you know that parabiosis is there, and you know the components, eotaxin or beta-2 microglobulin, then I think that in that case, yes, you can take the low levels of eotaxin from young people that is independent, or to work with the beta-2 microglobulin levels of young people, and then you try to cure everything. But I don't think that, yes, to do changes, transfusion of one to the other, no, this will be a disaster.
For a novel, maybe. Literature. Okay, thanks very much, Jesus. Okay. So we go on. Our next speaker is Carlos Matute, and the talk is entitled Contribution of Glial Cells to Neurodegenerative Diseases.
The main interest of our laboratory has been studying the impact of glial cells into neurodegeneration. And I would like to provide you with some information today, which is mostly unpublished, showing that glial cells can be primarily damaged in the neurodegenerative diseases. Specifically, I don't think it works. I am trying both ways without success.
Here for next.
Okay. Thank you. So specifically, I would like to show you three main ideas. The first one deals with the signaling of beta amyloid directly on astrocytes. The second about the effects of beta amyloid in oligodendroglia and myelin. And finally, I would like to show you the role of astrocytes in Parkinson's disease spreading. So there is an overall consensus that one of the major features of Alzheimer's disease is the aberrant processing of the beta, of the amyloid precursor protein. So in this, I cannot see the pointer, right? Well, anyhow. So oops. Sorry. So environmental factors, so genetic factors, result in the overproduction of amyloid species, and this, when released, can cause the accumulation in the extracellular space, which ultimately lead to the formation of plaques of different types. So these A-beta oligomers can be synthesized in the laboratory and assayed in different cell types. So we use this preparation of synthetic oligomers, which in a Western blot show that they are formed mainly by tetramers and pentamers. And when we look at the electron microscope, we saw that the majority of them have this particular rounded form, suggesting that they are mainly oligomers. And there are also a minority of fibrils, but the majority, which is the idea I want to emphasize when talking about this preparation, is composed by oligomers. So when we challenge initially neurons with these oligomers and record from them, we found that a beta amyloid would trigger currents which had the pharmacology of the NMDA receptors, as displayed here. And these currents would cause ultimately excitotoxicity. And we could evaluate the toxicity in areas which are particularly vulnerable to Alzheimer's disease, as the hippocampus and the entorhinal cortex. And what you see here in color is a marker which is propylium iodide, which intercalates into the DNA and shows which cells have the membrane damage and they are dying. And again, when you do the pharmacology of this cell death process, you see that it is mostly mediated by NMDA receptors, and since it is blocked by the specific antagonist to those receptors, and also by AMPA receptors. We wonder, after doing these initial experiments a number of years ago, what would happen in astrocytes. This worrisome about astrocytes is not new. Since more than 100 years ago, Nicolas Achoucarow, who was a pioneer in the study of glia at the turn of the last century, working with Alzheimer's, himself found that astrocytes form particular processes which surround the neurons. And this occurs again in vulnerable areas of Alzheimer's disease like the hippocampus. We very recently observed that these formations, when you use higher resolution microscopy, they are indeed present in transgenic mice, which I will talk later about. And when you go into the electron microscope, you see that there is a net surrounding the cell bodies, which seems to separate the synapses from that cell body physically by astrocyte processes. So we did studies of signaling with the oligomers I showed you before in different preparations in acute hippocampal slices, firstly isolated astrocytes, and also in primary cultures. In all of them, we saw that the oligomers would generate an increase in the intercellular concentration of calcium. By the way, the shape of the signal differed depending on the population of cells we were using, on the origin or the preparation we were using, but in all of them, such a signal was present. Elena Alberti, making a tour de force, pharmacological tour de force, trying to characterize those signals, found that the signal was not mediated by glutamate receptors nor by purinergic receptors, and that it was blocked by using blockers or antagonists of the inositol triphosphate receptors in the ER or the ryanodine receptor. In addition, and interestingly to us, she found that blocking integrin beta-1 would reduce greatly the signal. So, to make a story short, what activation of beta-1 integrin by oligomers does is activating the NOX2 oxidase, and this activation leads to an increase in the expression of the NOX2 itself, and in addition to the generation of reactive oxygen species, all of which are blocked by using a non-selective antagonist of this family of NOXes, or more specific antagonists of the so-called NOX2 oxidase. And this was done using a lot of beta amyloid, 5 micromolar, and you may question this and say, well, is this physiological? So to go a step further, these experiments, we produce native oligomers by transfecting N2A cells with the wild type APP or the APP with the Swedish mutation. And using this supernatant conditioned by these cells, and at picomolar concentrations, we found again that oxidative stress was generated in astrocytes. So I don't want to give you all the details about this signaling cascade. There is a poster dealing with it, and just to sum up, what A-beta oligomers do by interacting with integrin beta-1 is to activate the PI3 kinase, and all the cascade initiated at this level leads to the increase in expression of the NOX2 oxidase and the generation of reactive species, the increase also of the integrin beta-1 in the membrane, and ultimately to astrogliosis, and also to astrocyte cell death. So what about integrin beta-1 and astrocytes? Integrin beta-1 is expressed quite abundantly during development, but in the adult brain there is not much. Indeed, it is very difficult to locate it. You have to try many different antibodies. And what we found using one of those antibodies, that there was diffuse but prominent staining of the CA3 area and other hippocampal areas. And when we zoom in by doing electron microscopy, we found that indeed the integrin beta-1 was expressed in perisynaptic astrocyte processes. So it is possible that this integrin is playing a role, an important role, in the tripartite synapses, which is just there. In addition to perisynaptic astrocytes, we also saw expression in postsynaptic spines. So all those experiments, the ones I showed you briefly before, were done in vitro. So how about in vivo models? We used two options. The first one and the most obvious was to inject the oligomers into the hippocampus of a normal rat. And we found, again, that there was an increase in the GFAP, there was astrogliosis, and there was, again, an increase in the NOX2 levels. And when going to a more reliable model, the so-called triple transgenic mice, which recapitulates many of the major features of the disease, including the presence of plaques and defects in synaptic communication, we found that indeed in the hippocampus and other areas there was an increased expression in the levels of integrin beta-1, NOX2, and GFAP, the critical three molecular components of the pathway I am pointing out today to you. So, and this increase was more prominent in the older mice, so basically around 18 months. And when you compare the increase of those molecules with the levels of oligomers present in the sample, there was a clear correlation in all instances. So how about the histology? In histology, again, we're showing that there is an increased expression of all these markers around different types of plaques, like neuritic plaques here, or core plaques. So, and how about the EM? So we found that indeed astrocytic processes were abundantly marked with antibodies to the peptides, to the amyloid peptides. And this occurred around neuritic plaques and also around soma, which were undergoing degeneration. And when we look at the synapses, or around the synapses, we found that this peptide was also labeled, was present within these perisynaptic astrocytes. So it may well happen that this signaling and this accumulation of beta amyloid is impairing the tripartite synapse. But this is an idea, a working idea we have to work out, and we are planning to examine it by doing electrophysiology in the near future. So how about the post-mortem Alzheimer's brain? So again, the same thing. We find that integrin beta-1, NOX2, and GFAP is increased in average, at least. There is a subject heterogeneity, but in average there is an increase of the expression. And when we do the histology, again, astrocytes around the different plaques show an increase in the three markers, GFAP, NOX2, and integrin beta-1. So, and just to end up this block of experiments, we did a preliminary evaluation about the different stages of the Braak stages of Alzheimer's disease, and found that there is like a trend of an increase in as the disease is more severe in the expression of these markers. So to leave you just with a simple view of all these studies, it seems like as Alzheimer's disease progresses, there is an astrogliotic phenotype, which again advances, and this is driven, at least in part, by the increased abundance of amyloid peptides. I would like to briefly go to the other parts of my presentation, and the second one is the one dealing with signaling on oligodendrocytes. So this is a very simple scheme as to how an oligodendrocyte progenitor matures. You see, without going into the markers, we characterize the process that initially they have a very simple arborization and then a very, very elaborate one in which there is plenty of myelin and myelin proteins. And this is how it looks when you stain for them with those markers. So what we found is that submicromolar concentrations of oligomers increase the amount of myelin components, and that this increase could be attenuated or blocked by using blockers of the SRC kinases. And that experiment was done in dissociated cultures, but when you do those experiments in organotypic cultures, for instance, of cerebellum, you find exactly the same result. So it's not, this is a more reliable preparation than working in dissociated cultures, and it seems the same happens. So again, we have worked out the signaling of the oligomers onto the membrane of the oligodendrocyte. We don't know which is the receptor, in this case, to the oligomers, but we do know, and the details are provided in a poster, which we have here, is that there is an activation of the SRC kinase, and which in the PKA kinase, which drives the activation of CREB and ultimately leads to the increase of the basic myelin basic protein, which is a major component of the myelin. Interesting, we also found that the oligomers play a role in repair of myelin lesions. And without going into the details of the data, the experimental setup illustrates here that with the toxin, which is called isolecithin, you can demyelinate the preparation I showed you before, and then if you incubate the slice with beta amyloid, you get a better recovery of the myelin than if you do not. When again go to the myelin tracts of the triple transgenic mice, we see that regardless of the area we look in, corpus callosum or hippocampus, we have an increase in the levels of myelin. And when we do the histology, for instance, of the hippocampus, we see such an increase, which varies depending on the area we are in, and is particularly prominent in CA3, for whatever the reason. If we do the electron microscopy, we see alterations in the myelin in the triple transgenic. For instance, we see thicker myelin sheaths. There is hypermyelination. Indeed, we see changes in the appearance of oligodendrocytes, and we also see signs of de novo myelination. In there, we wonder what was the impact of these changes in the function. And I want to show you very, very recent data obtained last week, indeed, using a test. So in which we put the optic nerve of the transgenic mice or the control into a chamber, we stimulate it from one end and measure the so-called compound action potentials at the other end. And what we get is this kind of recordings, and we can evaluate the first peak when it is coming, the latency, and also the number, or an estimate of the number of actions which are activated. And what we see is that there is a reduced and impaired impulse conduction in the triple transgenic, which is quite marked. And the only question is, this is very preliminary, the number of experiments is still very small, it's 4 in each of these. So what happens in the human brain is the same thing again. There is signs, clear signs by Western blotting that there is an increase in the myelin content, as you see here, in the brain as compared to the controls. And when you do a preliminary study again of the different groups according to the BRCA stages, it looks like there is again a tendency to have a higher hypermyelinization as the disease advances. So, and just very briefly, the last part of my talk deals with another astrocytes, another property of astrocytes. In collaboration with the laboratory of Urban Besar in Bordeaux, we have the chance to get Lewy body extracts which were taken from patients with Parkinson and challenged them in neuronal cultures and astrocyte cultures. What we found is that after incubating for a few days, we could detect human synuclein within the neurons and also within the astrocytes. So, interestingly, neurons would suffer, the extract would be toxic to neurons, as you see here, but not to astrocytes. This is a very interesting feature. So astrocytes are resistant, but neurons are vulnerable, as expected. So the way the synuclein reaches the cell, the inner part of the cell, is through the endocytotic pathway, as you can see here, that they are with markers of endosomes or lysosomes, which are introduced. They are using baculoviruses. And this is also the same process in neurons and in astrocytes. So we wonder whether the synuclein containing those extracts would jump from one cell to another. And for that, Fabio Cavaliere, who is showing a poster with the details of this study, exploited these microfluidic chambers in which you can put one cell type in one side and another in the other, and then through channels, there is growth of processes, and you can load in one side the extract, the Lewy extract, and then put pressure at the other side so that if there is transmission from one side to the other, it would be through an active process. And what he found is that there is transport. If you put neurons here, from neuron to neuron, there is transfer from astro to astro, and there is also transport from neuron to astro and the other way around, as shown in here. So this means somehow that astrocytes can propagate indeed or suggest the pathology. So finally, when you incubate with the extracts, the different cell types, astrocytes and neurons, there is an increase in the expression of the endogenous alpha-synuclein, which is quite shocking and marked. Yes, this is the message I want to leave you with. Beta amyloid alters astrocyte function via intergene beta-1, and it targets oligodendroglia and causes hypermyelination, and astrocytes are resistant to pathogenic alpha-synuclein but facilitate disease spreading. And this opens plenty of questions. Let me just point out only a few of them. So the first one is, can markers of glial dysfunction, and in particular the ones I was mentioning, GFAP, NOX2, or intergene beta-1, can be used as surrogate markers of disease by exploiting, for instance, the availability of cerebral spinal fluid? We don't know. It's something we'd like to test. And we are undergoing conversations as to how we can do this effectively in a reasonable number of samples as a pilot project. So the second question is does beta amyloid pathology alters specific functions of oligodendrocytes, like the behavior of the tripartite synapse, or just the neurovascular coupling? We don't know that, but we are designing experiments to test specifically those points. And what is the impact of hypermyelinization in Alzheimer's disease? Does it affect the overall information processes? Does it contribute to the symptoms overall? We don't know. We have to do more experiments in the models we have. And finally, is the astrocyte syncytium a target for drugs attenuating the spreading of Parkinson's disease pathology? We don't know, but this is something we want to test, and we are on the way to test. So finally, to acknowledge the work of the collaborators, especially Elena Alberdi, who has led the work on beta amyloid signaling, and Fabio Cavalieri, who has done the experiments with the Lewy extracts. And also to our collaborators in our surrounding in the Achucar Center, José Luis Zugaza, Estíbal Capetillo Zárate, Erwan Besar, and Pepe Obeso, a member of CIBERNED. And I thank you for your attention.
Thank you, Carlos. I think we have time for some questions.
Only a short question about the pending question number 2 that you saw us already. I think that when you increase the amount of beta amyloid to neurons, beta amyloid could interact with receptors, NMDA or glutamate receptors. And the consequence is the death of the neuron. But it's different if those receptors are present in the synapses or in the extracellular part, in the extrasynaptic regions. Then my point is, if the synapsis is a tripartite synapsis that requires 2 neurons plus astrocytes, in that case, the receptors are in the synapses. But if you destroy the astrocytes, I don't know if it could go to the extrasynaptic part. And then will be the toxic thing. What do you think about this possibility for the pending 2 questions?
I think there are a number of options in there. So what is the source of the toxic oligomers? So is the presynaptic terminal? That's one option. Or is the astrocyte itself, which contains a lot. So in either case, I think the synaptic receptors and the extrasynaptic receptors, which are in the spine, would be affected. So those 2 possibilities are an option. So it can happen both ways. Maybe it happens both simultaneously somehow.
Thank you, Carlos, for a very nice presentation. I have 2 questions related to the effects that you see in myelin. As you know, one of the substrates of base 1 is myelin. So when you don't have base 1, you have overproduction or a lack of degradation of myelin. So I have 2 questions about that. I wonder if your in vivo data in the mice that they are overexpressing and the Swedish mutation, or as you know, is a better substrate for base 1, what is happening is that your base 1 enzyme in these animals is too busy cutting down APP and is not cutting down myelin. That's why you see an increase in myelin. Have you considered this? This is the first part of the question. And the second part of the question is more clinical, is regarding your results, what do you think about BACE inhibitors as a treatment for Alzheimer's disease?
I didn't get the second one, sorry.
The second one?
Yeah.
What do you think about BACE1 inhibitors as treatment for Alzheimer's disease?
Ben?
Base 1, beta secretase inhibitors.
Yeah. Well, the first one, so to us is a bit weird that we find what we find, so that there is hypermyelination, because our intuition would say that if there is hypermyelination, there would be fast. We didn't go in this kind of speculative thought, but it is a thought. I think it may be a byproduct of a number of abnormalities we find in the white matter tracts of these mice. It would be nice to check if this is the case also in the human brain. We cannot do more proper EM studies in the human brain, but it would be very important to do, although the fixation and the tissue preservation is not ideal to do that. And then the second question, so considering beta secretase inhibitors for that, we did not consider that option at all. So why do you think that would be, what would be the rationale to do that? Sorry for questioning.
Well, I was thinking, you know that you are aware there are base 1 inhibitors approaches for Alzheimer's disease. Since base 1 is the major enzyme cleaving myelin, if you inhibit this, you will find even more myelin, more myelinization, and this is what you want to avoid according to your data. So it will be a bad approach according to your data. So it was a little bit and this thinking. And about the first question, it will be good to see if it's the same result in a PS1 mice, a mice that is not carrying a good substrate for base, is carrying just more APP or something else.
Yeah, but that's a good idea to test. Thank you.
Thank you very much, Carlos. I'm afraid we have no more time for questions. Thanks. So we continue with our last speaker today is Jordi Alberch, and the title he's going to talk about is Neuronal Plasticity in Huntington's Disease, New and Old Molecules that Modulate Endogenous BDNF and P75 Imbalance.
Okay, thank you. And I would like to thank first the organizer for inviting me to give this talk and show you some of the results that we have in our lab. Today I want to show you what we are doing to try to identify new molecules, but really they are not new. So they are all molecules, but with a new effects that maybe can be useful for the treatment of Huntington disease. As you know, this is the progression of in Huntington disease in the patients, and there is a stage of neuronal death, but the early stage in a neuronal dysfunction. We have been working for maybe about 25 years in Huntington disease. And in the beginning, what we spent a lot of years trying to block the neuronal death. Maybe this was a mistake or something like that, because what could be useful is to go to the early stages and try to block or try to repair the neuronal dysfunction. So this is what we are doing here with the neuronal death versus neuronal plasticity. In the last year, we focused to try to identify the mechanisms that modulate the neuronal plasticity and neuronal dysfunction and try to find treatments for that. Well, you know that in Huntington disease, mutant Huntington is acting through so many different mechanisms in synaptic dysfunction, gene transcription, intracellular transport, mitochondria, autophagy, etc., etc. But we want to focus what we have been working with the trophic factors. With the trophic factors, we have been characterized the different neurotrophic factors that act on the different neuronal populations that are affected in Huntington disease, and really think that everybody agree that BDNF could be a very potent trophic factor for these molecules. BDNF can induce improve the synaptic function, the neuronal survival, and motor coordination, but it's also affecting the cognitive processes. And really, mutant Huntington is able to modulate the synthesis of BDNF, and also there is a reduction of BDNF. You can see here that BDNF is the most potent. So you see this is a wild type, it's a culture, striatal neurons, and when you treat the cells with BDNF, there is an increase in the branching, in the neural outgrowth, and also the survival is the most potent. There is not any other trophic factor with these clear effects. 10 years ago, more than 10 years ago, Lena's Cataneo group described that it was a reduction in the BDNF levels in several mouse models, but also in human patients. So this was the beginning. Together at this time, we also found that in different animal models, BDNF could be protective in the excitotoxic effects. Sorry. Well, we did a paper sorry, that this effect of BDNF was really important for the pathophysiology of Huntington disease. And what we did is to develop a mouse that they have mutant Huntington, but low levels of BDNF. So we cross-made the R6-1 mice together with heterozygote for BDNF. And here it was, they developed severe symptoms in the motor coordination in the rotor rod. And I skip this. And also we could see that the population that was most affected was the encephalinergic neurons. This population they are the first ones that are affected in Huntington disease. So these papers, this study really show that BDNF has a role in the pathogenesis in Huntington disease. If we, in another set of experiments, we try to overexpress BDNF in a mouse model of Huntington disease, and what we did is we have one mice that only overexpress BDNF under the GFAP promoter. So in these mice, they have normal levels of BDNF, but when there is any kind of pathology, cytotoxic insult, there is an increase of BDNF. What we did is to cross-mate with these animals with these R6-2 mice, and what we got is a double mice that have mutant Huntington, but overexpress BDNF when there is a pathology. Otherwise, it's normal. Here you see that this mice, you see that this is a mice with a mutant Huntington, and they have mutant Huntington, but with higher levels of BDNF. So that we could compare two animals with a mutant Huntington and the same number of repeats, but they have different levels of BDNF. In the previous one, they had the low levels, and this one we have high levels. And this was a protective effect. This is in the clasping, but also in the rotarod, we could see that was an improvement in these mice because they have higher levels of BDNF. So this was another evidence that BDNF can modulate and the onset and also the severity of the symptoms. And here the postsynaptic and presynaptic markers were higher. So these studies and some other studies show that BDNF was involved, but we could check also about the receptors. How are the receptors that bind BDNF? BDNF binds two different receptors with a different function. One is the TrkB that induces survival, axonal growth, and also cell proliferation. And there is another receptor that is P75. This is not only for BDNF, could also bind other neurotrophins, and they have the opposite effect. P75 is a cell death receptor and can induce an inhibition of the axonal growth. So this is the situation that we have with a BDNF that can bind two different receptors with a different function. One is inducing as a negative modulator, and the other is a positive modulator of the synaptic plasticity. And how are they in Huntington disease? These are two papers that Silvia Ginés did and Veronica Brito, and what we found here is that P75 is increased, and also Lena's Cataneo group also found the same results in different mouse models and also in human. And we could see that P75 is increased in Huntington disease, in mouse models and also in human patients. And TrkB is decreased. These are the differences. So there is an imbalance between P75 and TrkB. To find the functional effect of this upregulation of P75, what we did is to develop a mouse that we cross. This is the knock-in mice with heterozygote for P75. So what we did is to decrease the levels of P75, and when we decrease the levels of P75, we could find and recovery of the spatial and non-spatial memory deficits, and also we improve the dendritic spines that were downregulated in the knock-in mice. So we could find. So that means that P75 is also quite important to modulate cognitive and also the number of spines. So I mean synaptic plasticity. So if we know that BDNF could be relevant and also the activity of its receptors, and the next step will be to put BDNF in the patients or on the clinical trials. And the main problem is the way that we can do that. So BDNF is quite large. There is poor pharmacokinetics properties, and also, I mean, it doesn't cross the blood-brain barrier. So we have to identify new ways to deliver BDNF to the brain, and the best one could be to try to modulate endogenous BDNF and also the P75 TrkB imbalance. So even with exercise, there are some papers that show that BDNF can be increased, and good food and exercise can improve. But anyway, it's not enough, so we have to find another ways to do that. There are different papers that they try to do this pharmacological manipulation. We participated years ago in a paper with, we used cysteamine. We did this paper with a French group, with Sandrine Humbert and Frédéric Sardou. And this study opened a clinical trial to try to use cysteamine that was usually, it was used for a different diseases, and it was then used in Huntington disease clinical trial. And what I want to show you today is there are 3 drugs that we have been studying during these last years, and with different effects. They are all of them are focused in the BDNF and track B and P75 imbalance. And the first one could be the prostaglandins. And what we'll try to do is to stimulate endogenous BDNF. And the second one could be the hydroxyflavone, that to see if it's a real partial track B agonist. And fingolimod, that it could be a modulator of the P75 and track B imbalance. Okay, this is the first one. This is a prostaglandin signal transduction pathway that prostaglandins, as you know, has a lot of different activity, even in the vascular system, but they have receptors also in the brain. And there are different kind of receptors with different signal transduction pathways. And what we show you today is the EP2. So we started in a previous study that we did with EP1, and later on we did with EP2. That is what I want to show you today. This is the first study that really linked prostaglandins also with the pathophysiology of Huntington disease. And this receptor usually have a toxic effect in the brain. So what we did is to use an antagonist and try to improve the motor and also memory deficits in these mice. But if we go to the EP2, this the first point that why we use it is because the receptors are located in the brain areas that are really related with Huntington disease, the cerebral cortex, striatum, and hippocampus. And and there are also other papers that show that EP2 could be related with the secretion of BDNF. In these different papers, in different cultures, what they did is to find that prostaglandins can induce BDNF and also NGF, and also is this related with long-term synaptic plasticity. And then what we did is to use an agonist of the EP2 receptor, and we could find that when we treat the cultures, there is an increase in the number of neurites. We use a misoprostol. I don't know if you can see here the slide. And also another interesting point, and that it was an increase in the release of BDNF after the treatment. This effect in the culture was BDNF dependent, because when we use an antagonist of, an antibody that blocked the BDNF effect, the effect of the misoprostol was also blocked. So that means that this prostaglandin receptor activation, what induces is an increase of BDNF dependent effect. If we move to in vivo, we could see that this study was done now is to study cognitive effect, to study memory. So what we did is to study mainly in the hippocampus. This was interesting because this was a specific effect most in the hippocampus. This was not effect in the striatum and in motor coordination. When we measured with a T-maze or with a novel object recognition test, the NORD, we could see an increase and a recovery of the effect in the long-term memory after the treatment. we use mini pumps in these studies. These receptors are mainly affecting, or they have many receptors in the astrocytes, and we try to check if there is the markers of inflammation were also increased, but we could not see it. So there is an increase in BDNF, but in the other markers of astrogliosis or inflammation was not increased. So it seems it was more an effect specific of BDNF. When we did the effect in the presynaptic or postsynaptic markers, we could also be a recovery in these markers. Another interesting point is that there is a reduction in the effect of in the inclusions of Huntington. Well, this is the summary of the results that we got, and we could see that it's known that TP2 can induce through cyclic AMP and PKA to an increase in the phosphocrep, and also this could be induce a BDNF release that also can have the regulation in the neuronal plasticity. Okay, let's go to the second one. This is the hydroxyflavone. This is a drug that can be taken by a plant. And what we did is, it was a paper that showed that, described that this could be a good agonist, a potent agonist with an neurotrophic effect. And even what we also try is to identify the structure of this drug and to compare with BDNF and try to identify if we can make new drugs using the structure. And this is what they did in the beginning, but there was some problems that the previous results could not be reproduced for all the people. And then we spent a lot of time trying to see this really was an agonist of tract B, or maybe it was another mechanism that has protective effects. You see here there is not any band to try to phosphorylate tract B. But after a waste of time making so many experiments, what we found is the problem was the antibody. The antibody, there is a different residue that are in the tract B receptor to phosphorylate, and this is the 515 and the 816. And the main problem, what we could find in the next experiment, is that the hydroxyflavone only activates this part of the tract B receptor. So finally we could identify, and also this is interesting because this activates the PLC gamma pathway and not the other pathway. So what we found at the end was that this was a specific agonist of this 816 residue of the tract B receptor. So here we could see that there is a quite good activation of the 816 and not of the 515. With the Western blot, we could see also that this activation of the PLC, the PLC gamma. After the treatment with this drug, we could see an increase in the branching of the neurites. And also, we could find, when we did the treatments in vivo, that there is an improvement of the motor activity, motor coordination. This is mainly in the higher speed, and also with the novel object recognition test. So there is an improvement also in the cognition deficit. What is interesting with this molecule also is the better effect were found in these encephalinergic neurons and not with the substance P. So this is more similar one that we could find in Huntington disease. These are in R6-1 mice. And also there is a reduction in the number of Huntington aggregates. So the results may mean that we have a drug that could be a specific agonist of one specific and residual of the tract B and could phosphorylate only the PLC gamma receptor. And just to finish, this is the fingolimod, that this is a drug that is used for multiple sclerosis. So this is approved by the FDA. And this is an agonist of the sphingosine 1 phosphatase receptor and is a modulator of the immune system. And we found that this paper that really fingolimod could be also useful in a mouse model of Rett syndrome, and we tried to check if this could be useful for Huntington disease. And then we did this treatment. This is a long treatment with R6-1 mice. And we checked this study, what we did is the cognitive effect. There was also a previous paper that showed some results with a motor coordination. And we did here with cognitive effects in memory during the TMACE and the North. And the effect was quite clear with objective with a novel object recognition test, and also with the TMACE, there is a recovery after the treatment. Also with the number of spines, we check in the CA1 pyramidal neurons, and there was an improvement in the number of spines, and also with the PSD95 as a postsynaptic marker. Also, fingolimod is now that can modulate astrogliosis, and there is astrogliosis in there is an opposite way, and we could see that there is a decrease. I think that this is in the opposite way. So this is the these are changed. So this is the Huntington disease with astrogliosis, and this is the decrease after the treatment with the fingolimod. Also, we check the overactivation or NF-kappa B signal, and also that is found in the astrocytes, and also fingolimod could improve, attenuate this overactivation. What is interesting with this is that when we found that there is an increase after the treatment of BDNF, but we could not detect any changes in the BDNF protein. This happened several times because in the long-term treatments it's always difficult to catch where is the increase of BDNF. But we could see is that fingolimod treatment prevents the P75 upregulation in the hippocampus. You could see this, what we found in the beginning, that there is an increase of P75, and after the treatment, there is a reduction of P75. And also, this decrease that there is in the tract B, we could see also an increase after the treatment with fingolimod. So it seems that this drug is also modulating the P75 and tract B imbalance. We followed through trying to study the mechanism, and there is that this drug induce an increase in CMP levels and also promote CREP and raw phosphorylation that could explain the changes also in plasticity after the treatment. And another point is that this regulation of fingolimod could be induced by the modulation of TNF-alpha levels. When there is an increase in the TNF-alpha induce an increase in P75 that is reduced after the treatment. So in summary, this is the effects that we found. This was published very recently in the Human Molecular Genetics, and this was the cover of the issue that showed that fingolimod treatment can modulate the astrocytes, reducing the TNF-alpha, and also modulating the TrkB and P75 imbalance. And also we were very happy that this was also chosen as a new in the Nature and Reviews in Neurology about the possibility that is a drug that is already approved that could be used for the cognitive benefits in the patients with Huntington disease. So this is what we wanted to show you, that we have different molecules with different effects, and to try to find a trophic therapy maybe is not enough just to give BDNF. Also, we have to try to improve the P75 and TrkB imbalance. They are the people that did this last work with the fingolimod, and these are the group in the CIBERNED with the different PIs, Pep Canals, Esther Perez, and Silvia Gines. And thank you very much.
Thank you very much, Albert. I think we have time maybe for a couple of short questions. Sí.
Hi. So first of all, congratulations and thank you for your comprehensive review of all your extensive work during all these years, and particularly the latest ones. And now, to me, the most obvious question is how close to clinical trials are all these compounds that you mentioned, the prostaglandin, the dihydroflavone, and the fingolimod compounds? Are they facing clinical trials soon?
Well, now we are trying to speak with Novartis, who has this fingolimod, and also because now really we found that new approaches on these new functions for this drug, but I think it could be very close, because this is used for the multiple sclerosis and could be very easy to use. I think they are also used for different things. I mean, all these prostaglandins. I mean, with this we have to check that prostaglandins, they can do so many effects. So then we have to be careful that not to have any secondary effects for that. But I think that could be very good to try to get closer, because there is not any say damage in the beginning for this, because they are already used for different kind of patients.
Yeah. Jordi, considering the opposite effects that TrkB and P75 appear to have, is there anything known about the ratio of pro-BDNF versus BDNF, and related to that, could Huntington be involved in that modulation of the levels of these two forms of BDNF?
I don't know if you follow your question. You're asking, what is the level of BDNF that could modulate?
And pro-BDNF. So pro-BDNF would be acting more on P75. So is there anything known about the ratio of these two forms of BDNF in the brain, in normal brains, in pathological brains?
Yeah. Well, you know, pro-BDNF is very complicated, together with the P75. So the P75, they have so different effects and even contradictory effects. Another point that we have to check when we work with BDNF or possibility of the regulation could be the cleavage of BDNF could be a very good modulator to move this to BDNF or pro-BDNF for both of that. But we don't know what is the ratio that could be done. We always check also pro-BDNF in these animals, in these situations.
Jordi, I also would like to congratulate you, but I have a question. Since your main effect were found in the hippocampus and the most clear improvement was in memory, and you didn't find change in BDNF level, did you look at NGF?
Is what? Sorry?
Did you look at NGF?
NGF?
Yes.
We didn't check NGF, but I think it could be also a good point to, because in one of the papers that I showed here, at least with EP2, there was increase in BDNF and NGF, so I think that could be a good point also to check NGF and TRK that is also involved in this process.
Okay, thank you very much, Jordi. I would like to finish today thanking all the speakers. I think they did a great job, especially administrating their time during their talks because we are on time. So thanks very much to everybody. Yes, I have to remind you that the bus will be going out at 7:30, so please be there. Thank you.